day, class, and thank you for attending our lecture. So, for this day, we're going to tackle your lesson 3, cell migration and inflammation. So, you have your handouts. Please fill them up while the discussion is ongoing. And if you ever have any questions, do not hesitate to type it in the chat box. So, let's start. So, for this lesson, class, we're going to tackle your cell migration and your inflammation. So the first thing you need to take note of cell migration is how it would occur. Now, cellular movement would accomplish three main functions. The first one, class, would be the delivery of leukocytes of the myeloid lineage. When you say myeloid lineage, class, we're talking about the WBCs. Mainly yung neutrophil nyo and yung monocyte from the circulation. When you say circulation, we're talking about the peripheral blood into the tissue site of infection or injury where the cells would perform their protective function of eliminating infectious pathogens, clearing dead tissue, and repairing the damage. So to simplify this class, the first job of your cell migration is to allow neutrophils and monocytes to go to site of infection, allowing them to perform their duties and responsibilities such as eliminating infectious pathogen, clearing dead tissue, and repairing the damage. Now, the second purpose or the second function of your cell migration is the delivery of lymphocytes from their site of maturation. If it's a B cell, a B lymphocyte, it's from the bone marrow. If it's a T cell or a T lymphocyte, it's from the thymus. To the peripheral or the secondary lymphoid organs, specifically class yung lymph nodes, where the cells would recognize antigen, proliferate, and differentiate into effector and memory lymphocytes. When you say effector class, these are your cells capable capable of protective function. Capable of protective function. Basically, class, they could fight off the antigens. So that's the second function. They deliver your lymphocytes from their site of maturation, such as your bone marrow or thymus, to the peripheral secondary lymphoid organs, such as your lymph nodes, where they would differentiate. Pwede sila maging, they could become your T cytotoxic for the T cell, or they could become your T helper. Sa B cell naman, class, pwede silang maging memory, pwede din silang maging plasma cell. And the last function, class, is the delivery of these effector lymphocytes from the secondary lymphoid organ in which they were produced to sites of infection in any tissue where they perform their protective function. So yung mga activated lymphocytes na class. Once they reach the lymph node, di ba, some of them would undergo activation, if you still recall in the previous lesson. From their class, from the secondary lymphoid organ where they were activated, they will now go to the site of infection in any tissue where they would perform their protective function. So please copy. All right, let's continue. Now, let's talk about your leukocyte migration class. Now, leukocyte homing and recruitment to different tissues are governed by some general principles. Number one is that naive lymphocytes. Now, when you say naive, these are inactivated lymphocytes class. They would continuously migrate mainly into the secondary lymphoid organ, whereas lymphocytes that have been previously activated by antigen, known as the effector lymphocytes, as well as myeloid leukocytes, preferentially would home into tissues where there is infection or tissue injury. Memory lymphocytes class would migrate into lymphoid organs, mucosal tissues, skin, and other tissues. Okay, for you to understand this class, yung mga naive or the inactivated lymphocytes would continuously go to the secondary lymphoid organ. While those activated lymphocytes, also known as your effector lymphocytes, as well as myeloid leukocytes, that would go, ulitin ko class sulat ko, activated, activated lymphocyte, activated myeloid leukocyte, ito yung neutrophil nyo and monocyte, they would continuously go into the site of infection. Site of infection. So, ang mga naive or inactivated lymphocytes would go to the secondary lymphoid organs. Those that are activated naman, activated lymphocyte, activated neutrophil, activated monocyte would go to the site of injury. While those that are memory lymphocytes naman class would migrate into the lymphoid organ, mucosal tissue, skin, and even other tissues. Now, the second principle is that leukocyte homing and recruitment would require the adhesion of leukocytes to the endothelial lining of post-capillary venules, a process that involves molecules on the surface of both leukocyte and endothelial cell. So class, leukocyte homing for WBCs daw, for WBC to go to site of injury, for WBCs to go to site of injury, they must attach or they must perform adhesion. They must attach or they must perform adhesion to the endothelial lining. Ano ba tong endothelial lining na to, class? Itong endothelial lining nyo this is the one that surrounds your blood vessel. Dito yan. Now, in the blood vessel class, there are certain receptors. The same with your WBC. So, yung WBC nyo class, kunwari ito yung WBC nyo. On the surface of WBC, meron tayong mga tinatawag na adhesion molecules and chemokine receptors. So we have here your adhesion molecules, meron dito chemokine receptor. Now, for your WBC to attach to the endothelial cell, dun sa surface ng inyong mga blood vessel, itong mga receptors na to, itong mga adhesion molecules na to, they would also attach to the same adhesion molecules and chemokines found sa endothelial cell. So, para mas ma-appreciate nyo tong number 2, ito, take a look at this picture. Ito, this one. Ito yung sinasabi ko. Now, in the surface of your WBC, meron tayong mga tinatawag na adhesion molecules. Saka mga chemokine receptors. Now, sa chemokine receptors na yun, ito sila. Yan. Yan yung mga yan. Now, same din class sa mga endothelium or sa endothelial cell, meron din siyang mga adhesion molecules. Meron din siyang mga chemokines. Now, ang mangyayari class, kung ito yung endothelial cell, ito yung endothelium, meron yung mga receptor yan, magro-roll over ngayon yung WBC dyan. And yung mga receptor at adhesion molecule ng WBC mag-a-attach class. It would attach to the same adhesion molecules and chemokines found on the endothelial cell. So ulitin ko class, for your WBCs, balik tayo. For your WBCs to move or to go to the site of infection, they have to attach to the endothelial cell using their adhesion molecules and chemokine receptor. The third principle naman class is that endothelial cells at the site of infection and tissue injury are activated by cytokines secreted by sentinel cells in the tissue. Ano ba itong mga sentinel cells na to class? Sentinel cells class include your dendritic cell, macrophages, and your mast cells, resulting in the increased expression of adhesion molecule and chemokines. The consequence is increased adhesiveness of the endothelial cell for circulating myeloid leukocyte and previously activated lymphocyte. This naman class sa third principle na to, at the site of infection daw, dun sa site of infection, yung mga endothelial cells na to would have increased. Dadami daw. They will have increased expression of adhesion molecules and chemokines. And what causes this? This is caused class by the presence of sentinel cells. Dahil sa mga sentinel cells ng class, dadami daw yung adhesion molecules, dadami yung chemokines. And an example of sentinel cell class is yung dendritic cell, macrophage, and mast cell. So kapag dumami yung chemokines, the more chemokines present class, the more myeloid leukocytes and activated lymphocytes will go to the site of infection. The fourth one, because microbes and necrotic tissue induce the expression of the molecules that mediate leukocyte endothelial adhesion, effector leukocytes would migrate through the endothelium mainly when and where they are needed. So, regulated class ang pagpunta ng mga effector leukocytes na to. And this is controlled by the expression of those adhesion molecules. So kapag onti yung adhesion molecules, onti yung mga chemokines, onti lang din yung pupuntang mga effector leukocytes. So please copy. Okay, let's continue. Now, let's talk about these adhesion molecules na to, class. Now, ano ba itong mga adhesion molecules na to? Now, adhesion molecules are those that are of adhesion of circulating leukocytes to vascular endothelial cell is mediated by two classes of molecules. So, meron daw two class ng adhesion molecules. They are your selectin and they are your integrides, as well as their ligands. Now, the expression of these molecules were varies among different types of leukocyte and in blood vessels at different location. So let's talk about first your selectin and their ligands. Now, selectin's class are plasma membrane carbohydrate binding adhesion molecules that mediate an initial step. Please remember this, an initial step of low affinity adhesion of circulating leukocyte to endothelial cell lining post capillary venule. Meaning, class, yung selectin nyo is a type of adhesion molecule that only gives a low affinity of adhesion. Meaning, mahina lang yung pagkaka-attach ng WBC mo doon sa endothelial cell or sa endothelial lining. That's what your selectins do. Let me repeat, yung selectins nyo, class, these are plasma membrane carbohydrate binding adhesion molecules that mediate the initial step of low affinity adhesion, meaning mahina lang, class, very weak ang adhesion niya to the endothelial cell. Now, endothelial cells would express, please remember this, endothelial cell would express two types of selectin. They are your known as your P-selectin, known as your CD62P, and your E-selectin, known as your CD62E. Pakitandaan lang yung difference. Letter P for P-selectin and letter E for E-selectin. Now, P-selectin class so named because it was the first found in platelets. This is stored in the cytoplasmic granule of endothelial cell and is rapidly redistributed to the luminal surface in response to histamine. So, marirelease lang siya, class, kapag nakadetect siya or na-detect ng surface niya yung histamine from mast cell and thrombin generated during blood coagulation. So let me repeat, yung P-selectin nyo, class, was named P-selectin kasi una siyang nakita sa platelets. Now, they are stored in the cytoplasmic granule of endothelial cell and would only come out or redistribute in response to histamine from mast cells and thrombin generated during blood coagulation. Well, the second type of selectin naman, class, is your E-selectin. This is synthesized and expressed on the endothelial surface within 1 to 2 hours in response to cytokine interleukin-1 and 2 more necrosis factor which are produced by tissue sentinel cell, yung dendritic cell and macrophage nyo. So, ang pinagkaiba, class, ng P-selectin sa E-selectin, yung P-selectin nyo, class, would only come out in response to histamine and thrombin. Pag si E-selectin naman, class, they would come out in response to interleukin-1 and 2 more necrosis factor produced by yung mga dendritic cell nyo and macrophage. And this is usually seen in infections. Now, microbial products such as lipopolysaccharide would also stimulate E-selectin expression on endothelial cell. Now, ano ba tong lipopolysaccharide na to, class? If you still remember in your bacteriology, di ba meron kayong mga bacteria that are composed of lipopolysaccharide? And one of those is yung enterics nyo. So, ito lang yan, class. This is what you need to remember. P-selectin, class, released during histamine secretion from mast cell and thrombin and is seen in blood coagulation. Meaning, class, si P-selectin, lumalabas yan kapag may injury ka. While si E-selectin naman, class, would react in the presence of interleukin-1 and 2 more necrosis factor which are commonly seen in infection. So kapag meron kang bacterial infection. This would occur due to yung dendritic cell nyo in macrophage. And lipopolysaccharides, which are found in some bacteria, such as your enterics, can also stimulate E-selectin. Now, there is a third type of selectin, yung tinatawag nilang L-selectin or yung CD62L. Now, this is expressed on leukocytes. Hindi ko siya sinama, class, kasi yung selectin nyo, as I mentioned earlier, was only found on endothelial cell. Pero itong L-selectin na to, siya lang yung katangi-tangi na nakikita sa leukocytes. So please remember this. Pag tinanong kayo sa exam, what type of selectin is found on the surface of leukocyte? Ang sagot nyo, yung L-selectin. Pag tinanong naman kayo what selectin is found on the endothelial surface, ang answer nyo would be yung P-selectin and E-selectin. Now, this CD62L or L-selectin is expressed on leukocytes and not on endothelial cell. Now, L-selectin on neutrophils would promote adhesion of the cells to endothelial cells that are activated by interleukin-1, TNF, and other inflammatory cytokines. So, meaning class, si L-selectin nyo would be active on endothelial cells that are affected by E-selectin. So, ang mag-partner class is yung L-selectin at E-selectin because yung L-selectin nyo would be activated in cases of interleukin-1 and tumor necrosis factor, which, as you all know, as mentioned earlier, is in response to infection. Now, in adaptive immunity, L-selectin is required for naive T and B cell or lymphocyte to home into lymph nodes, meaning they are required class para makapunta for your B cell and your T cell to go to the lymph nodes through specialized blood vessels called your HEV. HEV class stands for high endothelial venules. So please copy. Okay, let's continue. Now, let's talk about the second one, yung integrin class and the integrin ligands. Now, integrins are cell surface proteins that would mediate adhesion of cells to other cells. So, kung ang selectin nyo class is found on the endothelial surface, ang integrin nyo naman would allow adhesion cell to cell. So, kung ang selectin nyo, wait lang, Integrin, cell to cell. Ang selectin nyo naman, cell to endothelial. Ang integrins nyo naman would be cell to cell or to the extracellular matrix through specific binding interaction with various ligands. Now, integrins would rapidly increase their affinity for their ligands in response to intracellular signals, which are induced in all leukocyte by chemokine binding to chemokine receptors and in T cells by antigen receptors. So nakakapag-increase sa class. They increase affinity. They increase the affinity or the attachment of cell-to-cell interaction. And so please come. Okay, let's continue. So this is the summary class of your major leukocyte endothelial adhesion module. So di ba merong family dalawa, selectin and integrin. So for the selectin, we have your P-selectin, E-selectin, L-selectin. Lahat yan class, si selectin nyo is known as CD62. Now, nag-iiba-iba lang yan if it's a P, it's an E, or an L. So lalagyan nyo lang ng, if it's a P-selectin, lagyan nyo ng P sa dulo. If it's an L molecule, lagyan nyo ng L sa dulo. So P-selectin or CD62P is found in the endothelium activated by histamine or thrombin. Si E-selectin nyo, endothelium activated by cytokine, specifically yung TNF saka interleukin 1. Si L-selectin nyo naman class found on cells, specifically neutrophil, monocyte, T cell, naive, and central memory, and even naive B cell. Now, ito naman yung ligand class. Now, yung ligand class, they help in the affinity or the attachment. So, for the P-selectin, ang ligand niya are of two types. You have your sialyl Lewis X on PSGL-1 and other glycoproteins. Now, they are found in this type of cell, neutrophil, monocyte, T cells, effector in memory. And then, ang E-selectin nyo naman class, they are found on glycoproteins, neutrophils, monocyte, T cell, effector memory. Yung L-selectin nyo naman class, sialyl Lewis X, PNAD on glycam-1, CD34, MADCAM-1 others, and your endothelium. Basically, class yung ligand. Yan yung opposite niya. Yung opposite na pinag-a-attach niya. So, kung ang P-selectin nyo at E-selectin nyo, both are found in the endothelium, ang ligand niya class would be the cell. So, in this case, itong sialyl Lewis X, nakikita sila sa cell, specifically sa neutro, sa mono, and so on. Kaya pansin nyo si L-selectin. Di ba si L-selectin nakikita niyo yan sa WBC? Ang ligand niya class are found on the endothelium. So, opposite. Kung saan nag-a-attach, ganun siya. Then, let's talk about your integrin. So, we have your LFA-1, CD11A, CD18. So, ito class medyo complicated but you have to memorize this. Then, we have your MAC1, CD11B, CD18. Then, we have your VLA-4, CD49A, CD29. So si MAC1 si LFA-1 nucleus are found in cells, neutrophil, monocyte, T cell. Kaya ang ligand nila class ICAM-1, CD54, ICAM-2, CD102 are found in the endothelium and upregulated when cytokine activated. Then yung MAC1 naman class, they are found in neutrophil, monocyte, dendritic cell. And ang kanilang ligand is ICAM-1, CD54, ICAM-2, CD102, and they are found in the endothelium, upregulated when cytokine activated. Then yung VLA-4 nyo, also known as CD49A, CD29, found in monocyte T cells, naive, effector, and memory. Ang ligand nila class is yung VCAM-1, CD106, and they are found on the endothelium, upregulated also, meaning the dami sila class when cytokine activated. Then we have your alpha 4, beta 7. This is known as your CD49D, CD29. Then we have in distribution, they are found in monocyte, T cell, naive effector, B cell. Now, ang ligand nila class is yung VCAM-1 and yung MADCAM-1. Found also in the endothelium sa gut and gut-associated lymphoid tissue. And then ito yung mga meaning class ng mga ligands nila and other shortcut names. So CLA-1 would mean cutaneous lymphocyte antigen 1. GlyCAM-1 would mean glycan-bearing cell adhesion molecule 1. Here have or high endothelial venule. ICAM-1 intracellular adhesion molecule 1 and IL-1 interleukin 1, LFA-1 leukocyte function associated antigen 1. MADCAM-1 is mucosal addressine cell adhesion molecule 1, PNAD peripheral node addressine, PSGL1 p-selectin glycoprotein ligand 1, TNF stands for tumor necrosis factor, VCAM-1 would stand for vascular cell adhesion molecule 1, and VLA-4 would stand for very late antigen 4. So please copy the table class as well as the meaning of those shortcuts. Okay, let's continue. Now let's talk about your chemokine and the chemokine receptors class. So chemokines are a large family of structurally homologous cytokines that stimulate leukocyte movement and regulate the migration of leukocyte from the blood to the tissues. The name chemokine is a contraction of the chemotactic cytokine. Basically class, ang chemokines nyo, kung ang cytokines nyo, yung selectin, yung integrin nyo, is for attachment, ang chemokines nyo class would be for movement. They movement and they regulate the migration of leukocyte. So there are around two major families and four subfamilies. We have your CC chemokine. So we have your CCL2. Ang original name ng CCL2 nyo class is yung MCP-1. The chemokine receptor niya is CCR2, mixed leukocyte recruitment. And then we have your CCL3. Ang original name niya is MIP-1 alpha. Receptor niya is CCR1, CCR5. Function niya is for mixed leukocyte recruitment. Then we have your CCL4, MIP-1 beta. Ang chemokine receptor niya is CCR5. Function niya is for T cell, dendritic cell, monocyte, and NK recruitment. It is also class an HIV co-receptor. Then we have your CCL5. Ang original name niya is RANTES. Its chemokine receptors are your CCR1, CCR3, and CCR5. It is responsible for mixed leukocyte recruitment. Then we have your CCL11, also known as eotaxin. Ang receptor niya is CCR3, responsible for eosinophil, basophil, and T helper 2 recruitment. Yung CCL17 nyo is also known as STARC or T cell recruitment, responsible for T cell recruitment. Ang receptor niya CCR4. Then yung CCL19, original name niya is MIP-3 beta or ELC. Ang receptor niya is CCR7. Function niya is for T cell and dendritic cell migration into parafollicular zone of lymph node. Then we have your CCL21. Yung original name niya class is SLC. Chemokine receptor CCR7. T cell and dendritic cell migration into parafollicular zones of lymph node. Then we have yung CCL22. Original name niya is MDC. Chemokine receptor is CCR4. Function is for NK cell, T cell recruitment. Then we have your CCL25. Original name niya is your TECK. Chemokine receptor is CCR9. Function niya would be lymphocyte recruitment into intestine. Then we have the chemokine CCL27. Ang original name niya is CTAC. Chemokine receptor is CCR10. Major function is for T cell recruitment into the skin. Then we have another group, yung CXC chemokine. So we have yung CXCL1. Its original name is GRO alpha. Chemokine receptor is CXCR2, responsible for neutrophil recruitment. Then we have yung CXCL8. Original name nito class is yung interleukin 8. Receptor niya is CXCR1, CXCR2. Major function is for neutrophil recruitment. Then we have yung CXCL9. Original name niya is yung MIG or yung MIG. Chemokine receptor is CXCR3. Then its major function is effector T cell recruitment. And then we have yung CXCL10. Original name niya is IP10. Chemokine receptor is yung CXCR3. It is an effector T cell recruitment. Then we have yung CXCL12. Original name niya is SDF1. Chemokine receptor is CXCR4. Major function is for B cell migration into lymph node, plasma cell migration into the bone marrow. Then we have yung CXCL13. Ang original name niya is BCA-1. Chemokine receptor niya is CXCR5. Ang function niya class is for B cell migration into lymph node and into follicles. T follicular helpful, it's also responsible class for T follicular helper cell migration into the follicles. Then meron tayong C chemokines. Under that is yung XCL1. Original name niya class is lymphotactin. Chemokine receptor is XCR1. Function niya is for T cell and NK cell recruitment. Then we have your CX3C chemokine. Under that class, you have your CX3CL1, also known as your fractalkine. Yun yung original name niya. Fractalkine has a chemoreceptor CX3, CR1. Major function is for T cell, NK cell, and monocyte recruitment. Yung CTL dito class refers to cytotoxic T lymphocyte, IL, interleukin, NK would refer for natural killers. So please copy. All right, let's continue. So let's talk about the biological actions of your chemokines class. Now, some chemokines are produced by cells in response to external stimuli and are involved in inflammatory reaction. Other chemokines class naman are produced constitutively in tissues and would maintain the distribution of cells in this tissue, such as the localization of T and B cell in lymphoid organs. Now, in inflammatory reactions, chemokines would serve to recruit circulating leukocyte from blood vessel into extravascular site. Now, chemokines would play two roles of inflammation. They increase the adhesion of leukocyte to the endothelium, and they cause the migration of leukocytes through blood vessel and toward the site of infection or tissue damage. So, yan yung two roles nila class kapag may inflammation. Increased adhesion and migration of leukocyte. Now, chemokines are involved in the development of lymphoid organs, and they regulate the traffic of lymphocyte and other leukocytes through different regions of secondary lymphoid organ. Now, chemokines are required for the migration of dendritic cells. They are required for the migration of dendritic cells from the site of infection into the draining lymph node. Di ba class ang dendritic cells nyo are also considered as antigen presenting cells. So, ang mangyayari dyan is that yung antigen or yung foreign substance, a-attach ngayon siya dun sa dendritic cell. And this would occur sa site ng infection. Now, from the site of infection class, pupunta siya ngayon sa lymph nodes. And sa lymph nodes class, it would now introduce, ipapakita niya ito, or it would now introduce tong mga antigens so that yung mga T cell, yung mga B cell would become activated. So, yun ang main importance niya, class. Bakit yung dendritic cells nyo kailangan pumunta ng draining lymph nodes. So, it allows the activation ng T cell and B cell. So, please copy. Okay, let's continue. Now, let's talk about, class, the leukocyte endothelial interaction and leukocyte recruitment into tissues. Now, leukocyte recruitment from the blood into tissues would require adhesion of the leukocyte to the endothelial lining of fossa capillary venules and then movement through endothelium and vessel wall into the extravascular tissue. So what happens is that there would be your selectin-mediated rolling of leukocytes on the endothelium. This would be followed by chemokine-mediated increase in affinity of integrins, stable integrin-mediated arrest of leukocyte on endothelium, and transmigration of leukocyte through the endothelium. Okay, class, ganito yan. If you still remember, sa discussion natin kanina, di ba ang selectins nyo would have a low affinity binding. So what happens here is that, ganito yan. Whenever there would be, sulat nyo to sa side ng handouts nyo. Whenever there would be a certain antigen, an antigen on that would invade the body, the antigen would invade the body, class, would invade the body. Macrophages would come and secrete secrete cytokines. Now, the cytokines, class, number 2. Itong cytokines na nasecrete ng macrophage, the cytokines secreted by macrophage would activate would activate the selectins. The selectins found on the endothelial cell. So, dadami yung mga addition molecules ng selectin sa endothelial surface. Now, what happens, the third step would be there would be increased blood flow. There would be increased blood flow. And when there is increased blood flow, class, this would cause WBC to migrate. Now, what happens here is that number 4, yung mga receptors found on the WBC would attach now to the selectin on the endothelial surface. After that class, after that attachment, integrin naman. Yung integrin na nakikita sa cell, integrin found on the leukocyte, found on the WBC, would attach, would now attach to corresponding receptors. So mag-a-attach yung integrin dun sa corresponding receptors sa in the endothelial cell. And when that happens, class number 6, isulat mo. The WBC would roll until it performs diapidesis. It would diapidize or pupunta ngayon magda-diapidesis siya towards number 7. Diapidesis towards the site of infection. Ganun ang mangyayari. So, ayan o. Nag-change shape siya, class, and it would now go to the site of infection where it would now go to the chemokines. Yung mga chemokines sa attach sa kanya. And it would now fight off the microbes. So, ulitin ko. Number 1, class, when an antigen would invade the body, macrophages would come and secrete cytokines. Now, the cytokines secreted by the macrophage would activate the selectins on the endothelial cell. The next thing that would occur, that would increase blood flow, causing your WBC to migrate. Now, receptors found on the WBC attaches to the lectin, specifically yung selectin na ligands nyo. Now, when that happens, class, integrin found on the WBC would also attach to the receptors found on the endothelial. Dito ka na magkakaroon ng increased affinity. Mas malakas na yung attachment mo. When that happens, the WBC would now roll and it would perform diapidesis. Diapidesis would be toward the site of infection. So, yun ang more steps ng movement ng inyong WBC. This is how yung WBC nyo, class, would migrate. So, please copy. Okay, let's continue class. So this is the migration of neutrophils and monocytes to site of infection. So after maturing in the bone marrow class, neutrophils and monocytes would enter the blood and would circulate throughout the body. Now, although these cells can perform some phagocytic function within the blood, their main function, including phagocytosis and destruction of microbes and dead tissue cell, would take place in extravascular site of infection. Basically, class, this is virtually anywhere in the body. Now, blood neutrophils and monocytes are recruited to tissue sites of infection and injury by a selectin, integrin, and chemokine-dependent multi-step process. Yun yung in-explain ko kanina, which follows the basic sequence common to the migration of all leukocytes into the tissue. Neutrophils are the first type of leukocyte to be recruited from the blood and into the tissue of or the site of infection or tissue injury. So ang unang WBC class na pumupunta kapag merong infection or tissue injury, yung neutrophil nyo. Now this would be followed by monocyte, which would occur hours later and would continue perhaps for days after neutrophil recruitment. Furthermore, in some inflammatory site, neutrophils are not recruited at all but monocytes are. These different migratory behaviors likely reflect various variation in relative expression of adhesion molecules and chemokine receptor on neutrophil versus monocyte. So itong last statement, class, there are some cases na merong mga neutrophil, there are some cases wherein neutrophil are not recruited but monocytes are. Now, the reason for this, class, is because of the variation, meaning iba-iba yung dami, iba-iba yung amount, or there is a variation in the expression of adhesion molecules and chemokine receptors on neutrophil versus chemokine. So, minsan, class, mas madami yung monocyte chemokines compared with neutrophils. Kaya there are cases wherein neutrophils are not recruited to the site of injury. So please come. All right, let's continue. So let's talk about this time, class, the migration of T lymphocyte. So lymphocytes are continuously moving through the blood, lymphatic vessels, secondary lymphoid organs, and non-lymphoid tissue, and distinct population of lymphocytes which show different trafficking patterns to the site. So kanina, class, if you still remember in the earlier slides, sabi ko yung mga naive lymphocytes would always go to the lymph nodes. Now, when you say naive, yan yung mga inactivated. So, naive T cells would preferentially leave the blood and would enter the HEV. Now, ano ba tong HEV na to, class? Ito yung high endothelial venule. So, kunwari, class, itong nakikita yung arrow. Yung arrow na yan yung blood vessel. Now, itong cell na to, this cell is yung naive T cell nyo. Yan yung naive T cell nyo. Now, yung naive T cell nyo from the blood vessel class would go to the high endothelial venule. Yan yung entrance class. Yan yung entrance ng naive T cell nyo papasok sa lymph node. Now, kanina class, sabi ko about dendritic cells. Di ba? Ang dendritic cells, they are required or they are necessary to go to the lymph nodes. Kasi di ba nga, ang dendritic cells nyo, they are considered antigen-presenting cells. They carry antigen or yung mga remnants ng antigen and they would help in the activation ng mga T cell nyo. So once the naive T cell, kapag yung naive T cell nyo class enters the lymph node through the high endothelial venule, yung dendritic cells class, ito sila, yan yung mga yan. Dendritic cell bearing antigen would also enter the lymph nodes through the lymphatic vessel. Now, if the T cell, yung mga inactivated T cell nyo would recognize antigen, ma-activate sila. So, dun nyo class makikita yung importance ng dendritic cell. By having the dendritic cell class, ito sila, ito yung dendritic cell. By having the dendritic cell also enter the lymph node, it activates the naive T cell, ito yung naive T cell nyo, to become an activated T cell. Now, itong activated T cell nyo naman class, once they are activated, so from naive T cell, magiging activated sila, they would now return, babalik sila class sa circulation. Ang daan nila class, they would pass through the efferent lymphatics. So, ito yan. They would pass through the efferent lymphatic vessel, thoracic duct, then into the superior vena cava, then into the heart, and ultimately into the arterial circulation. Now, meron ka na ngayong mga activated T cell. This activated, also known as effector T cell or memory T cell, would preferentially leave the blood. Aalis naman ngayon sila class. They would leave the blood, itong arrow. They would leave the blood and they would enter the peripheral tissues through venules at the site of inflammation. So pupunta sila dito class sa mga peripheral tissues niya. Recirculation through secondary lymphoid organs other than lymph nodes would also occur. So ganun nangyayari. So ulitin ko, naive T cell would enter the lymph node through the HEV or the high endothelial venule. Now at the same time or even in the previous pwedeng nandun na yung dendritic cell. Now si dendritic cell, they bear antigen. And it would cause the activation of your naive T cell, causing the naive T cell to become an activated T cell. Si activated T cell nyo naman would leave through the efferent lymphatic vessel, would go through the thoracic duct, to the cephalic vein, superior vena cava, and into the heart, and into the arterial circulation. Now, from their class, it would enter the peripheral tissue through venules at the site of inflammation. Then, it would also occur recirculation through secondary lymphoid organs other than lymph nodes. So, pwede siyang pumunta sa other lymph nodes class. So, please copy. Pag tinanong kayo class sa exam nyo, where does inactivated T cell enter the lymph nodes? Ang sagot nyo would be high endothelial venule. Pag tinanong kayo, where does activated T cell exit the lymph node? Ang sagot nyo would be efferent lymphatic vessel. Okay, let's continue. Now, we're done with the migration of T cell naman class. Next naman would yung B cell naman. So, naive B cells would use the same mechanism as do naive T cell. So, same lang din sila class. Naive B cell class would also go to the, yung naive B cell nyo, would also go to the lymph nodes. And they would also enter through the HEV or high endothelial venue. Now, what happens here is that once they enter into the lymph node via the HEV, naive B cell class would migrate into the follicles. Naalala nyo pa yung primary and yung secondary. Di ba kapag sa primary, this is where you would find inactivated B cell. Kapag yung secondary, ito yung may mga germinal centers where they would become activated na yung mga B cell. So, naive B cell would migrate into the follicle, to the primary follicle. B cell activation and generation of plasmablasts and memory B cell would occur. So, once na maka-encounter siya class ng antigen, maka-encounter siya ng antigen doon sa follicle mo, magkakaroon ng germinal center. And that is where activated B cell would be activated. Now, once the B cell is activated, there would be B cell activation and there would be the generation of plasmablasts and memory B cell. So, ulitin ko class ha, they have similar migration ng inyong T cell sa B cell. So, what happens is yung naive B cell nyo would go to the lymph node via the HEV. And once they're class, they would go to the follicle, specifically yung primary follicle. Kasi dun yung inactivated B cell nag-stay. Now, once they are able to detect ng antigen itong follicle na to, they would now develop germinal centers. And the follicle would now be known as your secondary follicle. And sa secondary follicle class, that is where there would be B cell activation and generation of plasmablasts and memory B cell. Now, once the plasmablast is created, or the memory cell is created, plasmablast and memory B cell would exit from the lymph node. And from the lymph node class, pupunta sila ngayon sa blood. Now, from the blood class, there are two things that would occur. Si plasmablast nyo would either migrate into the bone marrow or into the mucosa. And there are two types of plasmablasts. May dalawang klaseng plasmablasts. We have yung tinatawag na IgG plasmablasts and yung IgA plasmablasts. Si plasmablast nyo class na IgG plasmablast, sila yung pupunta sa bone marrow. And once in the bone marrow class, it would now secrete the type of antibody known as your IgG or immunoglobulin gamma. While another group ng plasmablast, si IgA plasmablast, would migrate into the mucosa. And in the mucosa class, it would secrete IgA antibodies. So, nagets nyo, di ba merong 5 types ng antibody? GAMED. So, si IgG plasmablast would go to the bone marrow. Si IgA would go to the mucosa. Kaya nga, class, si IgA, yan ang antibody. Please remember this. This is the antibody found in mucosal surfaces. Mucosal surfaces. So, yan ang pattern ng migration ng lymphocyte. Now, aside from that class, you have to remember that the movement of B cells from secondary lymphoid organs would depend on S1P. Follicular B cells in the spleen migrate to the marginal zone and then are carried by fluid through the red pulp and into the circulation. Now, S1PR1 deficient follicular B cells have diminished ability to leave the spleen. Subsets of B cells committed to producing particular types of antibody would migrate from the secondary lymphoid organ into specific tissues where they differentiate into long-lived plasma cell. So aside kasi class from the lymph nodes, yung B cell nyo kasi would also go to the spleen. Sa spleen class, they would now go to the marginal zone and they would be carried by fluid through the red pulp. Then from the red pulp, pupunta siya sa circulation. So ulitin ko, aside from the lymph node, si B cell nyo pupunta rin sa spleen. Now sa spleen class, they would now go to the marginal zone. And from the marginal zone, they would go to the red pulp and then into the circulation. Now, meron tayong tinatawag na S1P. Now, S1PR1 deficient follicular B cells have diminished ability to leave the spleen. So, kapag deficient sila sa S1PR1, hindi sila basta-basta nakakaalis sa spleen. And then from the spleen class, they would go to different tissues. In the case of your IgG plasmablast, pupunta ngayon siya sa bone marrow. Si IgA, pupunta siya sa plasma cell. So that's how migration of B cell would occur. So please copy. So familiar sa inyong class yung mga CCR7, CXCR4, yan yung mga receptors niya class, yung sa mga chemokine. So that helps in the migration of your ano. So makikita mo dyan yung how they would migrate through the help of those chemokines. So anyways, let's continue. Now let's talk about your inflammation. Now this is the principal way by which the innate immune system would deal with infections and tissue injury is to stimulate what you call acute inflammation, which is the accumulation of leukocytes, plasma protein, and fluid derived from the blood at an extravascular tissue site of infection or injury. The mechanisms that are activated in response to an infectious agent would constitute what is commonly referred to as an inflammatory response. Either tissue damage or infection can initiate the series of events that would lead to inflammation. Now, the four cardinal signs class of inflammation would include your redness, also known as your rubor, your swelling, also known as your edema, your heat, also known as your calor, and your pain, also known as your dolor. Now, meron din tayong tinatawag na loss of function. Hindi to lagi sinasama class kasi hindi naman all the times kapag may inflammation ka, nagkakaroon ka ng loss of function. So this is known as your functiolastia, loss of function. Now, major events associated with this are increased blood supply to the infected area. This would cause redness, increased capillary permeability caused by retraction of the endothelial lining. This would cause your swelling. Migration of white blood cells from the capillary to the tissue. This would cause heat. And migration of macrophages to the injured area would also cause heat. So please copy. All right, let's continue. Now, other events that would occur during inflammation class. So we have your chemical mediators such as histamine, which are released from injured mast cell, would cause the dilation of the blood vessel and bringing additional blood flow to the area, resulting in redness and even heat. The increased permeability of the vessel would allow fluid in the plasma to leak to the tissue. This produces swelling and pain associated with inflammation. Soluble mediators including acute phase reactant would initiate and control the response. So kapag increased permeability pala class, there would be swelling, swelling and even pain. So aside from swelling, dapat meron din tayong pain dito sa number 2. Then, as the endothelial cells of the vessels contract, neutrophils move through the endothelial cells of the vessel and out into the tissue. Neutrophils are attracted to the site of injury or infection by the chemotoxins. Neutrophils, which are mobilized in within 30 to 60 minutes after the injury, are the major type of cell present in acute inflammation. Neutrophil immigration may last 24 to 48 hours and is proportional to the level of chemotactic factors present in the area. Migration of the macrophages from surrounding tissue and from blood monocytes would occur several hours later and would peak at 16 to 48 hours. Macrophages attempt to clear the area through phagocytosis, and in most cases, the healing process is completed with a return of normal tissue structure. So please copy. Okay, let's continue. Last one, class. Chronic inflammation is a process that takes over from acute inflammation if the infection is not eliminated or the tissue injury is prolonged. It usually involves recruitment and activation of monocytes and lymphocytes. Chronic inflammatory sites would also often undergo tissue remodeling with angiogenesis. Now, when you say angiogenesis, class, this would refer to the creation of new blood vessel, as well as fibrosis, new fibers. Now, although innate immunity stimuli may contribute to chronic inflammation, the adaptive immune system may also be involved because cytokines produced by T cells are powerful indicators of inflammation. So please remember that, class. Yung mga cytokines produced by T cells are powerful indicators of inflammation. Yung fibrosis, class, ito yung thickening. The thickening or in some cases yung scarring ng tissue. Diba pag nagkasugat ka, there would be some sort of wound or scab na yun. So please copy. Alright, so that ends the lesson 3 class for your Immunosero, cell migration and inflammation. So you have until tonight to submit these handouts.